occurs in this female, and if we assume in this case that it occurs in meiosis 1, then all of the gametes are either going to be this XX type or the null type. So that's kind of an example of what would happen in this particular case here. We could propose it's this one too, in which we'd have more possibilities for the gametes, but the illustration goes through an example using non-disjunction in meiosis. Okay, so the possible gametes from the female are shown here. For non-disjunction, we line them up on this side of the Punnett square. The male we assume is gonna undergo meiosis correctly, and so the gametes are either an X and a Y, and we combine everything together, and we get certain output. Okay, so one progeny will be XXX. The problem is there's too many X's here. The gene dosage is too high, gene expression will be too high, and so we'll have development that's deregulated and will derail at some point, and so those embryos won't survive. So having too many X's in Drosophila isn't a good thing. 3 of them won't work. This is in contrast to, let's say, those sex chromosome disorders we saw in humans, where we have this inactivation system. In Drosophila, we don't have the same kind of inactivation system, and so this generates a chromosome imbalance that's lethal. So that's one. If XX combines with Y, we get an offspring which will have white eyes because there's 2 copies of this W allele. The Y doesn't do anything, so this is going to be a female that has white eyes. If we have the null chromosome here combining with X, we get this individual which has 1 copy of X, and in the Drosophila system that causes a male. Okay? And the male will have the W plus, and that will be red-eyed. And finally, if we have the null chromosome, or the null gamete here, plus this Y, we generate an organism that doesn't have an X, and that's also lethal because there's lots of genes on the X, and you have to have those for normal development. So this is kind of the output that's depicted in the event of a non-disjunction of the X chromosome in a Drosophila female in meiosis 1. Okay, so these would be the outputs. Now this example isn't restricted to the sex chromosomes, and it's not restricted to either the males or females. It's just an example set up in the book involving this Drosophila red-eye, white-eye model. Okay, you can have non-disjunction of sex chromosomes in the male. That's possible. You can also have non-disjunction of chromosomes that are autosomal, right, like trisomy 21 and things like this. Those are non-disjunction events as well. So all those things are possible. They work pretty much in a similar way. It's just that we're using this as a model to show how the breeding experiment works. So I've summarized this also in the next slide. Here's the mating experiment. We have a female that's XW, XW, a male XW plus Y, and what happens is the XW, XW collectively fertilizes either the X or the Y chromosome from the male, or you get nothing fertilizing either the X or the Y chromosomes from the male. And so our output is XW, 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 too many X's, okay? Or we get no X plus a Y, too few X's. So both of those will be lethal. The viable ones are XW, XW, and Y. So 2 X's makes a female, and they have white eyes because you have a W and a W, the homozygous recessive. And the other possibility is nothing from the female, but you get the XW plus chromosome from the male. That makes another male. They will have red eyes, but they will be sterile. And the reason they'll be sterile is that during meiosis, there's a kind of set of checkpoints, and one of the checkpoints is to make sure that the chromosomes have got things to line up with during the synaptonemo complex formation. And in this case, you don't have a Y or another X pair for this chromosome to line up with, and meiosis then thinks something is wrong in terms of chromosome numbers, and rather than proceed and make gametes that have the wrong numbers of chromosomes, we'll stall out right away. And so these animals tend to be sterile because the X doesn't have anything to pair up with. Okay, so that's a summary of what happens here. I do want to point out that if you have a population of female flies of this genotype and male flies of this genotype, you can't tell them what to do. You can't say, okay, please show us what happens in a non-disjunction event in meiosis 1 with this with an X chromosome and so forth. These are very rare events. It's not like all the population is undergoing this type of non-disjunction and has this type of progeny output. It's very rare. So really, in an experiment like this, it would be more, as I've described here. You set up a mating between Drosophila females that are homozygous, small w, versus males that are XW plus Y. What should happen, and what normally happens, is correct segregation of chromosomes during meiosis. So XW combines with XW plus, XW combines with XW plus, or XW combines with Y, XW combines with Y. Most of the offspring are fine, and in that situation you get half of the offspring being red-eyed females, that is XW plus XW plus, where W plus dominates over W, and half of the population of the offspring would be white-eyed males, and that would be XW plus Y. So you'd see hundreds or thousands of Drosophila progeny in this little bottle with this mating that you've set up, and they would look like this, and they would look like this. It's just that once in a while, you'd see something weird, okay? And the something weird would be a female that was white-eyed instead of the expected red-eyed, or maybe once in a while you would see a male that is red-eyed instead of the expected white-eyed, right? So these would be fairly rare offspring that would be due to very infrequent non-disjunction events, in this case involving the X chromosome in the females. Okay? So this is more typical of the actual breeding experiment, and then people would follow this up and say, well, what in the world is going on here? This isn't right. And they do lots and lots and lots of other experiments, and finally they come up with a model that correctly explains why they see these very rare offspring, and that model involves this non-disjunction. Okay, so that's non-disjunction. The last thing for this particular lecture is just to point out there's a private research foundation called Howard Hughes Medical Institute, HHMI, and it was started by this guy, Howard Hughes, who was a kind of an eccentric Hollywood mogul, and he ran a big aircraft company, so forth, and then he donated a lot of money to this Howard Hughes Medical Institute, and they give grants and so forth to usually young investigators who are very prominent and so forth. So they have a lot of money to give for people to do research. And on their website, they have a few interactive tutorials or things that you can go through which are kind of interesting. And this is an example of one of them. I think they've changed a little bit the format of it, but you can find it on this HHMI website if you want. And it has to do with gender testing of female athletes. And so this is kind of related to what we're talking about with respect to mating type, sex determination, and so forth. And their little interactive tutorial is pretty interesting because they describe a set of possible tests that you can use, whether it's detection of bar body or hormonal tests, things like this, and show how those tests have changed through the years. And they also describe how these Olympic committees and so forth have used various criteria for determining whether females can compete or not compete from, let's say, the 1960s up through current days. And the rules have changed all the time as well. So it's an interesting little thing. You don't have to go through it, but I think it's kind of a neat thing, kind of expands your horizons about the complexity of some of these issues. Okay. So good, that's kind of basic stuff on chromosomes and cell division and all that stuff. What we're going to do is now turn to something called extensions of Mendelian analysis. And so we'll look at this idea that genes come in multiple allelic forms. So far, we've seen, let's say, big S, big Y, small y, things like that. But genes come in lots of different allelic forms, and this has a consequence in terms of phenotypic output. And we're also going to look at a variety of 2-gene systems. The 2-gene systems we've looked at so far involve things like S and Y genes, where the genes control completely different things. But a lot of times genes control the same trait, right? They coordinate with one another to produce a particular output. And we're going to see lots of examples where our out of 16 Punnett squares give us a variety of phenotypic ratios because of the way the genes interact with one another. So that's kind of our goal in this lecture and this section. Before I go into that, I want to make a point about the new edition of the book. The new edition of the book combines extensions of Mendelian analysis, which we're going to talk about today, with a topic, non-Mendelian genetics, okay? The old book has separate chapters for those two things, which I think is a better way to do it, but nonetheless, that's what we have to deal with. So I just want to point out that if it happens that you have the new edition, keep in mind that this chapter is composed of 2 different parts, and we're going through one part today. The other part about non-Mendelian genetics we'll talk about after the first exam. Okay? So in chapter 13 of the new book, there are various sections about gene expression, environment, maternal effect, extranuclear inheritance. That means things like mitochondria. We're going to talk about all those things and more a little bit later in the course. Right now, we're going to focus on the topics that I'm going to discuss in this lecture. So make sure you kind of partition this chapter in the new book into these 2 categories. Okay, so first is just a real simple review of some of the genetic symbols that we might see. Already we've looked at capital letter alleles and small letter alleles as a way to designate the wild type version or the functional version versus a mutant version or a non-functional version, right? And we've seen that with big S, small s, so on and so forth. Sometimes you'll see a letter with a plus indicating it's functional and usually the wild type most common variant or allele of a gene. And the other one will just be a small letter by itself with no plus, which indicates that it's the mutant version, usually the recessive version, usually the non-functional version. So that's a way to describe different alleles. Another way is to forget about the letter altogether, but just indicate a plus. This assumes that you know what gene you're talking about, and the plus just is in a shorthand way of saying that's the good version, and then you designate specifically the mutant or recessive or alternate version by a small letter. So that's a way to depict dominant and recessive allelic forms. In bacteria, you often see something that describes the nature of the gene. So this is Lu plus, Lu minus. Lu plus would mean it's an allele of a gene involved in leucine metabolism that's functional, probably dominant, and Lu minus means it's probably the recessive or the non-functional version. So you see this used in bacteria frequently. And an example we're going to look at today, IAIB, as depicted here, means neither of these alleles are really dominant to one another. They're going to be codominant. You get an output from both of them. So we'll look at some of these, this system in just a second. Okay, so topic 1, multiple alleles. Turns out it's usual that genes come in lots and lots and lots of allelic forms, not just big S, small s, as in these Mendelian cases, okay? So if you have a chromosome as depicted here, and somewhere along the length of the chromosome at some spot, you have this nucleotide sequence, this chemical sequence that represents some gene, typically, at that spot, you've got lots of possible sequences that can be kind of inserted there on either the maternal or the paternal chromosome. So in this illustration, you have one allele which is designated big A, which is the wild type and probably dominant version that could be right here. But there's lots of possible sequence variants or alleles that might be present at that position, depending on the frequency of these sequence variants in the population. So we have allele A1, A2, A3, A4, and here they're trying to depict that there is a distinct mutation or nucleotide sequence change at different positions in each of these distinct alleles. And anybody could have either this one or this one or this one or this one or this one on one of their chromosomes, depending on the frequency of these in the population. So this is the typical situation. You know, if you think of 100 nucleotide sequence, right? Imagine 100 nucleotides is a gene. Now that's too short, but imagine you have a sequence of 100 and it's an exact sequence. If you just pick one location, maybe there's a G there. At that G, you could have 3 possible substitutions or changes, right? You could have, instead of the G, which would be wild type, you could have an A or a T or a C. Each one of those would be, let's say, an allelic variant or an allelic form of this hypothetical 100 nucleotide gene. And across every one of those 100 positions, you would have a chance of 3 possible changes. And each one of those would be an allelic version. So you can imagine, excuse me, there's lots and lots and lots of possible alleles, even for something as small as 100 nucleotides. And it doesn't have to be just one change. You could have 2 changes, right? That makes it even more complicated. So there's a lot of sequence variation in genes that might be present at a given locus. Yeah, question? Oh, you're talking about this? Okay, so I'm going to go over this. I assume colored means it's a slightly darker shade of pink where you're starting to approach red but not quite at red. Okay. So in this excuse me, in this table, you have something really interesting. What they've done here is they've taken female Drosophila of various genotypes, and they've measured the amount of dye, red dye, that's present in the eyes of these animals. And this explains why W plus is dominant to small w. So if you have a female which is homozygous W plus, it's going to make a certain amount of red dye. And if you measure that, we can set that at a value of 1, which is what they've done here. So the wild type W plus allele present at 2 copies is set at a value of 1 in terms of this dye or this pigment in the eyes, okay? So what about W, which is recessive and defective for pigment production? Here's a female which is WW, recessive and homozygous for the W allele. And you see that the amount of dye is very, very little in the eyes of these flies. Therefore, they're white-eyed flies, not red-eyed flies. And what you have in the rest of them is a whole bunch of possible alleles of this W gene, not just W plus, not just W, but there's lots of these. And in each of these cases, there's a slightly different amount of pigment production that will be diagnostic for the type of allele. So each of them is producing a protein that has the ability to produce a little bit, excuse me, but not nearly as much as the wild type does, okay? So the one at the bottom, I'm assuming, if this is 1 and this is 0.16, I would assume it's kind of pink, but a little bit stronger pink and going towards the red side. Okay. So this is neat. It tells you how much dye is produced. It also raises the question of, well, what about a fly that's W plus W? Why is that fly red-eyed? Why do we say W plus is dominant to W? So if the fly is homozygous dominant W plus, you expect equal production of pigment from an output of this chromosome and from this chromosome. So this value of 1 is really divided into 0.5 from one chromosome, 0.5 from the other chromosome, right? Each of these alleles is producing a protein, which is producing pigment, and they're functioning equally. If you have 1 copy of W plus and 1 copy of W, you would expect to produce about a level of 0.5 pigment from your 1 copy of W plus, and you would expect to produce almost nothing in terms of pigment from your small W allele. So a fly that would be heterozygous for W plus W would probably make a relative level of pigment at about 0.5, okay? Well, that's intermediate, but it just so turns out that to our eyes, a level of pigment of 0.5 compared to 0.1 isn't really distinguishable. They both look red to us, and that's why the W plus looks about the same as the W plus W, homozygous dominant versus the heterozygote. Okay, so the point is multiple alleles, a lot of possible sequence variations could be present at a given locus or a given spot for any given gene. And this is something that we'll look at again when we look at hemoglobin variants, for instance. Okay, second topic is codominance. For codominance, we illustrate this idea using this blood group genetic system, which probably many of you have seen, but just as a reminder, there are 4 different blood groups in this ABO blood group system. There's other blood group systems, but we're going to look at this ABO blood group system. And so a person can be O, they can be A, they can be B, and they can be AB. Okay? And it turns out what you are depends on the alleles you have at the I gene. And there's 3 possibilities. You can have IA allele, IB allele, and small I. Depending on what collection of alleles you have, right, and you can only have 2, one on maternal, one on paternal. Depending on what collection of alleles you have, that's going to determine what your blood group is going to be. And what these things do is they specify the addition of carbohydrates on the surface of red blood cells. So these IA and IB proteins have some catalytic activity that cause differences in carbohydrate addition. So in words, everybody has something called an H antigen on the surface of their red blood cells. It's a particular carbohydrate structure. If you have the IA allele, it converts the H antigen to the A antigen. If you have the IB allele, it converts the H antigen to B antigen. If you don't have either of those, if you're small I, then you don't produce any activity and H stays as H. And I've got a picture of that in the next slide here. This is the H antigen, or it's a carbohydrate group on the surface of red blood cells. The IA allele produces a protein that will convert this to this. This is the A antigen, so something has been added, so that's fine. The IB version of the I gene will act on the H antigen as a substrate and will go this direction to add this on the surface of the H antigen to produce the B antigen. So you have H converted to A with IA allele, you have H converted to this B antigen using the IB allele. And if you're small I, you don't do this, you don't do this, you stay right here. Okay, so that's kind of the genetic system. It's 1 gene, 3 alleles. If you have both IA and IB, you get both A and B. That's why they're termed codominant. They both will function. It's not one will kind of override the other. All right, so if you're genotype small I, you only make H and you're type O. If you're either IA small I or IA, then you're type A because you make A antigen. If you're IB/small I or IB, then you can make the B antigen, and so you're type B. And if you have both the IA allele and the IB allele, then you can make both A and B, and so you're type AB. Yeah, question? So the RH is a different blood group system. There's a variety of these blood group systems, okay? So they all have their own kind of genetic basis. You have to look up the detail. There's RH, there's MN, there's a variety of these things. This is just one of them. Okay, so that's the system, 3 alleles. This is how it works in terms of carbohydrate addition. But we have to understand something else in terms of the medical consequences of these different blood groups. And what we have to consider is the effect of the immune system. So during development, the immune system, which is really remarkable, is able to be trained to know what is self, what is you, and what is not you, what is foreign, right? That's why you're able to fight off infections or respond to things, because your immune system knows what should be, antigenic for yourself, and it avoids that, and it knows what is kind of foreign things, viruses and whatnot. So your immune system can make antibodies against things that are non-self, but it doesn't make antibodies usually against things that are self. So I've got two cases. Let's suppose an individual is blood type O. If they're O, they have small I. They have to be homozygous. They can't have either IA or IB. Right? So we know the genotype in the event of people being type O blood. If you're type, if you're type O, you make H, but you don't go here, you don't go here. H will be self for you, right? But you don't make this and you don't make this. So the A and B antigens will be non-self. So you have the ability, good or bad, to make antibodies or to mount an immune response against A and B blood types. So that's why you can't give transfusions of blood to people who can't accept that type of a blood. If you give, let's say, type A blood to somebody who's O, that A antigen has never been seen by a type O person before, and their immune system will mount a response to that. So if you're type O blood and you're given blood from someone with red blood cells that are A or B, your immune system either has seen that and has antibodies, or will make a response when it first encounters those A and B antigens. Okay, case 2. What if you're AB blood type? In that case, you have H, right? Some of the H is going this direction, some of the H is going this direction, so you have all three of these antigens, and so your immune system has been trained to think that all three of these are self, so you will not mount a response against this or this or this. So in the case of type A, B blood, you would not have antibodies against H or A or B because those are self, and so you could accept blood from any type. And of course, everybody knows that this ABO blood group system is important in terms of donating blood and receiving blood and so forth. And I want to point out that one of the reasons, obviously this is important because you mount an immune response, but one of the reasons that this is important medically is what I've indicated in this last paragraph. The three alleles, IA, IB, and small I, are present at relatively high frequencies in populations. Okay, so you don't know what a given person is going to be. So this is an important kind of aspect of this system. High allele frequencies for each of the three alleles makes it uncertain what blood group a person is. If one of those alleles was one in 10 million people, probably you wouldn't worry about this very much in terms of this blood group system and blood donation. But the fact is, you just don't know. There's an appreciable proportion of the population that will be O, that will be A, that will be B, and be AB, and so you have to worry about the system. Okay, so what happens in terms of the blood? I've already mentioned about this mounting of the immune response. A person that is type O can potentially have or will make antibodies against A and B. If they receive O blood, nothing happens. But if they receive A blood, these antibodies will coagulate or cluster these cells together and kind of make a mess. Likewise with B and likewise with AB. On the other case, the second case, AB, an individual with AB blood type will not make antibodies against A or B or H, and so they could receive blood from any of those types. So you've got a lot of components to this. You have a genetic system with three alleles. You have then a definition arising in terms of what type of blood group a person is, and then you have this kind of medical consequence in terms of blood donation type of procedures. So the last thing in this model, so what do you do with it? You can make simple genetics problems with these blood group systems, and here's one that's really, really trivial. They used to be used a lot. They're not used so much anymore because you can do DNA analysis and you can find things unambiguously, like parentage and stuff like this, but it's still a way of trying to evaluate relationships between individuals. So here's an example, really simple. A child that has blood type O, you know that they have to be small I, right? That's a given. So what could the parents be if the child is type O? The parents could be small I, or O themselves. They could be A, or they could be B. If they're A or B, they have to be this genotype, right? Or this genotype. They would have to be able to contribute a small I to generate a child that was small I. So any parent with the genotype that has at least one small I is possible as a parent, a legitimate, real parent of an infant that was type O. Okay? You can rule out one blood type, that is IA-IB. A parent who is IA-IB would have to give either IA or IB to their offspring, and so the parent could not be this if the offspring is small I. Okay. Also, the parent couldn't be homozygous for IA, nor could they be homozygous for IB. But this isn't necessarily easy to tell, because you can't tell the difference between a person of blood type A with this genotype and a person of blood type A with this genotype. Okay. So anyway, these types of problems, they're kind of fun. You'll see a few in the book, and they give some crude indication of the likelihood of parents matching babies and this kind of thing. The blood group system is an example of codominance. That is to say, if you're IA, you produce one output. If you're IB, you produce another output. And if you're both, you get both outputs. Okay, so they're kind of codominant. One isn't dominant over the other. So example of codominance. Okay, and then the last thing here for this blood group is a kind of puzzle. We have a woman who is not type A, and she's not type B. Well, if you're not A and you're not B, you have to be O, right? That's the logic and the system that we've discussed so far. So seemingly she's type O, but there's a problem. One of her parents was AB, but we just looked at this. If a person is O, if a baby is O, the parent cannot be IA, IB, because they have to transmit either IA or IB. And yet in this case, this puzzle, it was determined that one of her parents really was type AB and had IA and IB alleles, even though she seems to not be A or B and is O. And it was also determined that she passed on IB to two of her kids. So let's assume these things are all correct, right? They were determined and it's accurate. She transmitted IB to two of her kids. If she has IB and transmits it to her kids, why isn't she type B? So that's kind of a puzzle, right? So perhaps she got IB from one of her parents, she has IB, she passes it to her kids, all that makes sense. What doesn't make sense is why is she type A, why isn't she type B, at least, okay? And the explanation is she was carrying a mutation for a gene called FUT1, and FUT1 acts right here. FUT1 makes H antigen. Okay? So she's homozygous recessive for an allele of the FUT1 gene that's defective. So she can't even make H, right? So now this is gone altogether. So if she has the IB allele, that's fine and good. It should produce B antigen from H, but H isn't there as a substrate in the first place. So even though she may be producing IB as an enzyme that can catalyze this reaction, there's no substrate for it to work on, and so you don't see any consequence of having it. She can transmit it just fine, but biochemically it's kind of useless because she doesn't make this. Okay, so this is a puzzle, so-called puzzle called Bombay phenotype, and it's due to this mutation in the FUT1 gene. And then the question at the bottom is, what about her ability to receive a blood donation? Sorry? So what happens if she receives type B? She has the IB allele, right? But the IB allele produces a protein. That protein doesn't do anything. She never makes the B antigen, right? Yeah, but but I suppose that that population is pretty small, right? Yeah, so I don't know about that. Can you filter blood in some way to remove an antigen? I don't know, maybe you could enzymatically change it. I don't know. It seems like a possibility, but I don't know. But basically the take-home message is she's in trouble, right? I mean, she can't receive blood from most people who will have H antigen or A antigen or B antigen. Probably the only way she could receive blood from another person would be if they also had this FUT1 mutation, but that's exceedingly rare, which is why this is a puzzle in the first place. Yeah, question? Type O does not have A and it does not have B. That's right, that's good. But it has H. Okay? She doesn't even make H. So for her, this kind of central position, H antigen, is still foreign to her because she's blocked right here. Yeah. So it's, yeah. I don't know what it does, whether it's important or whatever. You'd have to ask a red blood cell. Yeah. Yeah. Yeah, so she could give it because she has no antigens whatsoever, in a sense, right? A or B or H. So she would be great as a donor, but receiving it, she's never seen any of those things, and so that would be the problem. But you know, she's just one person. How much she can't give that much. Okay, good. Yeah, so again, that's kind of a clinical thing, and I don't know. I suppose that you might be able to suppress her immune system transiently while she was receiving a donation, and then after that was done and that blood was gone and she didn't mount a response because she was suppressed, then she would be good again. So this might be a way around it. I mean, that's just kind of standard pedigree analysis. It must have just so happened that both of her parents were heterozygous, right? Let's see. One of her parents is IA, one of her parents is, she has to have gotten those from her parents, right? One defective copy of FUT1 from one parent, one defective copy of FUT1 from the other parent. Those parents presumably had one good copy, and so they were able to make everything here. Does that make sense? The other way, you could imagine that there's a spontaneous mutation. Something just so happened to her FUT1 gene during development, right? But that usually is so rare that you don't always invoke that kind of accidental spontaneous mutation. Okay, I need to move on to oh, here's distribution of IA allele worldwide, for what it's worth. I don't know. It's just information, right? I think what they do here is they try to measure the IA allele using some sort of DNA or PCR approach, and they try to do it with more or less endogenous populations, right? Some sort of a pre-migration type of populations, and you get some idea of the distribution of this allele. What is the meaning of this? Are there any theories? I don't know, just kind of a picture of how the abundance of this allele maps across the world. Okay, another example, incomplete dominance. So here we have an example involving chickens. They're Andalusian chickens, and we know the answer, and so the book is going to set up the problem in a certain way to kind of reveal what we know about this incomplete dominance type of model. So we start out with the chicken. This is a hen who has black coloration. There's a rooster, which is white. And we know the answer to this, and so they're telling you already that the hen is containing a coloration allele called CD, and it's homozygous. We know the answer, and so they're telling us that the rooster has this a variant allele at this coloration locus called CW. So probably this produces an enzyme that makes a pigment. Probably this produces an enzyme that's defective or broken or doesn't produce any. Okay. So if you have two copies, you're dark in color. If you've got two copies of this CW, you don't produce pigment, and so you're light. we line up the possible gametes on the Punnett square, and we get an organism that's intermediate in color. There is one copy of CW, which is producing pigment, another one copy of CB, which is producing pigment, one copy of CW, which is not. And so it stands to reason that the amount of pigment that you can produce here is 50% of the amount here when you have two copies of this. Okay? So the point here is that there's incomplete dominance. The situation with the Drosophila eye color was one where if you had a relative level of pigment of 1 and a relative level of pigment of 0.5, it still was observable to us as red eyes. In this case, having half amount of pigment produces a chicken that has an intermediate color. So this is a blue Andalusian chicken. And they will not breed true. Individuals that are heterozygous, if you breed them together, you're not going to get the same thing out again. And so if we cross the two heterozygotes, the two blue Andalusian chickens, if you suppose that you can make a strain of blue Andalusians, it won't work. Right? You get either of two possible alleles from this chicken, either of two possible alleles from this chicken, you line them up, you generate all the possible gametes that you get here. One part is going to have CBCB and will be dark. Two parts will be CBCW. They'll have half the gene dosage of that CB allele, and so they will be this kind of intermediate gray color. And one part will be CWCW, which will be white. So you get a 1 to 2 to 1 ratio. They don't breed. Oh, and this is why we're studying this, right? To make chickens and eggs. That's what they're. For better or worse, that's okay. So incomplete dominance. So the Drosophila eye color is complete dominance visually, but really it's incomplete dominance as well if you talk about the measurement of the pigment from the eyes themselves. Sorry? Right, in Drosophila example, the phenotypic output is 3 to 1 because the heterozygote shows dominance and recessiveness as we expect. In this case, the kind of genetics is the same. You've got heterozygous against heterozygous, and so you get 4 boxes in your Punnett square, but you have to apply different rules to generate the phenotypes, which gives you a 1 to 2 to 1 rather than the 3 to 1. Okay, another thing to discuss is this idea of complementation. So in complementation, as depicted here, we're going to talk about 2 genes which contribute to some phenotype, whatever that is. And so I have case 1 and case 2. In case 1, I have a parent here, and I have a parent here, and they're going to generate offspring here. And the same for case 2, parent and offspring. Okay? In case 1, the idea is this individual is homozygous for a mutation in gene 1, but they're normal for gene 2. And you need both gene 1 and gene 2 good copies in order to generate the correct phenotype. Okay? If you have a broken copy of gene 1, you get some disorder or some trait. Okay? So parent 1 is defective for both copies of their gene 1. Parent 2 also has this trait, this disorder, but the reason is because they have 2 copies of a mutation in gene 2. And gene 1 and gene 2 are both required to generate wild-type phenotype, whatever it may be. If you're defective for gene 1, you have the disorder. If you're defective for gene 2, you have this disorder. So these 2 individuals have the same phenotype, the same disorder. And yet, when they have offspring, they contribute, this person will contribute a bad copy of gene 1 and a good copy of gene 2. This person will contribute a good copy of gene 1 and a bad copy of gene 2. And so now you have an offspring with this genotype, heterozygous at both genes. The good copy overrides the bad copy. The good copy overrides the bad copy. This individual is normal. So this person is affected with some disorder. This person is affected with the same disorder. The offspring are perfectly fine. This is the idea of complementation, where having one good copy kind of overrides the other one. On the other hand, here we have a similar situation where this person has the same disorder because of a mutation in gene 1. This person has the same disorder. It's also a mutation in gene 1, but it's at a different location in gene 1, a different allelic form. The offspring that will be produced here, you can only get a broken copy of gene 1 from this person and a broken copy of gene 1 from this person. There are different types of broken copies, different types of alleles, but both of them will still be broken in the offspring. So here you have no complementation. So affected parent, still affected offspring when the mutations were in the same gene. Here you have affected parent, but the offspring are okay because of complementation. Yeah, question?
How would you know that the good gene would override the
So
mutated gene?
you don't always know that, but that's a kind of typical rule of thumb in the types of things that we're seeing here, right? Typically, when you talk about dominance and recessive in the types of examples that we've seen so far, dominance or wild type means the gene is producing a good protein product, an enzyme or whatever it is that's functional. And the recessive or mutant copy, the broken allele, is very often a defective version. So if you have something that works together with something that doesn't work, the thing that works still works. So that's kind of the logic for a lot of these. Yeah, so this is a question for population genetics and how quickly you get rid of defective alleles. Yeah, so if it's dominant and defective, it goes away faster. Yes, because if you're seriously affected with something and it's dominant, I mean, you're going to be inviable in some way, right? Those don't rise to that high level in the population in the first place, though, right? Because, yeah, for recessive disorders, it's very hard to get rid of bad genes. And we'll talk about this kind of topic when we look at population. Yes, right, and that's often true, and we'll see examples of this in the next few slides, where more than one gene contributes to the same phenotype. The Mendelian situation is really special and unique, where you have an S gene that does one thing and Y gene that does something completely different. Most of the time, genes work together to collaborate to produce output. So this is far more common than in Mendelian. Yeah, last one, then I gotta go forward, yeah. Yes, so you have to, so in this case, why does this work when you have one good copy, one bad copy? And the reason is, even though you only have 50% amount of protein production here for gene 1, let's say, compared to this, it's still enough. Is that your question?
It only works if that 50% of working.
Yeah, so for some genes, this kind of heterozygous situation, for a lot of genes, this kind of heterozygous situation will be perfectly fine. That's why diploidy is important and this dominance recessiveness works, okay? There are some genes when you have this situation where the 50% is not enough. So that can happen, but the typical situation is having 50% for most proteins, usually you're still okay. Yeah. Okay, so I gotta go forward. Now we're gonna look at some 2-gene systems. We've already looked at one 2-gene system in those Mendelian problems where we looked at S and Y. The problem is S controls one thing, Y controls something else. They're completely separate. Okay, so you can deal with the output of S and independently deal with the output of Y and just kind of bin them together in the various categories. So that's fine and good. It's very simple. A lot of times you have 2 genes, and those 2 genes collaborate with one another to produce proteins that affect some developmental process. So the 2 genes will interact with one another to generate more complicated phenotypes. And so we're going to look at some examples of these. When we look at those 2-gene systems from Mendel, we generate these Punnett squares with 16 boxes, right? And the phenotypic output in the case of Mendel was 9 parts, 3 parts, 3 parts, and 1 part. And you get that because we apply certain rules about the genes do separate things, the alleles are dominant and recessive, and all that. But you've got 16 genotypes in there, 16 boxes. And if you apply different rules, you can get different ratios of phenotypes out of a Punnett square with 16 boxes. So we're going to look at some 2-gene models where we get 16 boxes in the Punnett square, but we're going to apply different rules and get different phenotypic ratios. So let me see if I've said that right. Okay, so good enough. Let's go forward. Here's an example involving chickens again. And we know the answer to this. So the book sets it up in a certain way with parents of a certain genotype so that we get F1s of a certain genotype and then can get F2. So it's a little bit artificial, but it's set up in such a way to kind of illustrate what we think is going on. So this is a 2-gene system, and it involves a phenotype having to do with chicken combs, okay, which we're all interested in. Okay, so here we have a hen. Is it a hen? Yeah, it's a hen, and we're given the genotype in the problem so that we can lay it all out in this illustration. And the genotype is dominant at R, recessive at P. And as a consequence of having this allele combination, you get rose comb. Okay? The other, and this will be true breeding, right? The only possible gametes would be big R and small p. So true breeding. This would be true breeding also, but it's a slightly different collection of alleles at the R and P genes. In this case, it's recessive R and dominant P. However R and P are interacting, whatever those proteins do, having these two different combination of alleles as a genotype causes a change in development of the comb to look, in this case, like rose, in this case, like P. Okay? So now the gametes here will be big R, small r, big P. We can only combine them in one possible way to generate F1s, which are going to be heterozygotes. All the problems always laid out in this same way, right, in the book. We're always generating these F1 heterozygotes. Then we breed the F1 heterozygotes against one another to generate an out of 16 Punnett square type box. Now, in this model, we're still going to say that these chromosomes sort and segregate independently. So what that means is that we can treat the output of the R gene separately from the output of the P gene. So we're going to say, if we did a cross of this versus the same genotype, we would get 3/4 of the offspring being big R something and 1/4 being small r small r. So that's kind of the same thing that we did in those Mendelian problems as well. We treat the R gene first, and we can treat the P gene separately. So of the progeny of this F1 cross that are big R something, which is 3/4, 3/4 will be big P something, 1/4 will be small p, small p. Okay, so that's the same thing we did before. In this cross, 1/4 of the offspring will be small r, small r. Of those, 3/4 will be big P something, 1/4 will be small p, small p. So we do cross 1 with the R gene, and then we combine it with cross 2 for the P gene. And we can do that because we're going to say these are on separate chromosomes, and they're going to sort, segregate, and all that independent. So what do we get? We get a 9:3:3:1 ratio of genotypes. 9 parts dominant at both. 3 parts dominant at R, recessive at P. 3 parts recessive at R, dominant at P, and 1 part recessive at both. But now we don't apply the same type of rules about the genes control separate traits. We have to understand that some way biochemically these things interact together to produce a different developmental outcome. So it turns out if you're dominant at both, you get one particular look, which is the walnut look. If you're dominant at R and recessive at P, you get rose. If you're recessive at R and dominant at P, you get this P cone. And then finally you get the kind of the classic chicken look, right, which is recessive at both. This is called single. Okay? So it is 9:3:3:1, but you have to be careful and realize it's not exactly working the same way as those Mendelian problems where the 2 genes controlled completely different things. Here the genes are collaborating and in some way affecting the development of comb in a complex manner depending on which collection of alleles you have. So this is 9:3:3:1, but it concerns the same trait. Okay, so some pictures. Take a moment to admire these magnificent birds. Okay, here's rose comb. I don't know. Okay, walnut cone, yeah, it kind of looks like a walnut, right, a little bit. Pea, I don't know. And then this is the classic, foghorn, leghorn type of cartoon chicken. These will be easy to breed true, right? It would be easy to imagine you end up kind of at this point. All your chickens after that are always going to have this single type of a cone because you can only contribute small r and small p. Okay, just a note about these. It's not like you can look at these two gene systems and know what will happen. A lot of work is behind these things, so don't think that you can't figure it out necessarily just by looking. All of these different two gene systems involve the same type of 16-box Punnett square, and yet the rules we apply will vary in case to case, and you'll get different outputs. So it's very complicated. A lot of work has gone into these things. We already know the answer. That's why we're setting it up in a particular way to begin with, to generate F1s and then to generate F2s. So they're complicated problems and not something that you should just necessarily intuit immediately. So we see lots of situations where parents with kind of generic genotypes, A and B, for example, like this, are crossed to generate heterozygotic F1s, cross them again to generate 16-box Punnett squares, and the ratios are all over the place. So just keep that in mind. Another example, we're talking about squash here. That's also pretty exciting. So here's a squash, which is kind of an oval and yellow. And we already know the answer. The book is laying this out so we can follow it. But let's suppose that this parent squash has a generic genotype, homozygous dominant A, homozygous recessive B. And for whatever underlying biochemical reason, these two alleles combine with one another during development to generate this output. If you bred this squash against itself, it would be true breeding. You would only get big A and small b, and you'd produce the same thing generation after generation. The other parent is here. This is also a true breeding strain, but it's small a, big B, and it produces the same thing. So however these alleles are interacting with one another at the biochemical level, whatever the proteins are doing, the developmental output is the same here as it is here. You breed this against itself, you get the same thing. You breed this against itself, you get the same thing. Here, we're showing two squash that seemingly are identical, and yet, lo and behold, when you breed them together, you get something completely different and completely unexpected. And these puzzles obviously attract people's attention, and they want to figure out what in the world is going on here, right? So here we have a heterozygote F1, big A, small a, big B, small b. As a consequence of having this particular arrangement of alleles at the biochemical level, something happened differently than in this case to cause the production of a squash that's kind of disc-shaped and green. And as before, we take the heterozygotes, cross the heterozygotes. We're going to treat the A gene and the B gene independently when we do the cross. And so 3/4 of the offspring will be big A something. Of those, 3/4 will be big B something. 1/4 will be small b, small b. Of the offspring here that are the 1/4 that are small a, of those 3/4 big B something, 1/4 small b. So again, we get a 9:3:3:1 ratio of both dominant, one dominant, the other recessive, or one recessive, the other dominant, or both recessive. That 9:3:3:1 is the same, but we're going to apply different rules that don't match the Mendelian rules. In this case, for whatever reason, if you have at least one dominant A and one dominant B, you turn out to be disc-shaped. If you have a dominant A gene and are recessive for the other, or if you're recessive for the first and have a dominant B gene, you turn out to have this phenotype. Okay? And if you're recessive at both alleles, something else different happens during development to cause the production of yet another phenotype, which is this kind of elongated squash. So you still have an out of 16 set of genotypes in those Punnett squares, but we're applying different rules, not necessarily even knowing what the rules are, but different rules apply, and the output is different depending on what collection of alleles you have. So here you have 961 as a ratio of out of 16, not 9331 anymore. Okay, and I point out here that we could have started with big A, big B times small a, small b, small b. We still would have gotten this, but I think the result maybe is a little bit less dramatic if you do that, and so maybe they've just set it up in this way. But it's a bit arbitrary as to how you set up the parents as long as you get heterozygous for the F1s. Okay, so this one, 961. And again, these are not easy. You can't just know the answer in advance because you have to know kind of what rules you're supposed to be applying in given situations. Okay, so the last one I'll talk about today has to do with mouse coat color. And the coloration of fur in mammals has been a long-standing genetic model because right away you can see the result. You don't have to do any kind of invasive test to determine the phenotype of the offspring. You just let them grow up and you can look at them. So that's easy. And so people know a lot about the genetics of fur color because it's so easy to screen or to score the offspring. Okay, so to understand this situation of mouse coat color, we have to consider 3 genes. And they're listed here. One is a gene for agouti, and agouti produces a little yellow band on the fur. And at this agouti gene, we have 2 alleles to start with. Big A causes a yellow banding pattern, and small a does not. And big A is dominant to small a. Okay, so that's kind of one actor in this model. Another one is a C gene, and C gene comes in 2 allelic forms. Big C allows coloration of the fur and is dominant. Small c does not allow coloration of the fur and is albino. And then we have yet another gene, which is B, and it comes in 2 allelic forms. Big B causes black fur, small b causes brown fur. So we have to consider all of these things when we think about this mouse coat color genetic model. Okay, so the types of colorations are agouti, albino, black, brown, and yellow. And I've got some, first of all, this is a picture of what that agouti allele does. It makes a little yellow stripe in the fur at some very specific developmental period. So solid fur, if it's black, would look like this. An agouti trait on top of that black fur would look like this. And I guess the idea is to make the mouse more camouflaged or something like that. So here's some examples. This mouse is agouti. It's kind of intermediate. It's not a solid color. And you can see, I don't know what the MD stands for here, but the A means agouti, so you can see that the fur color is slightly yellow. This is also an agouti mouse. It's still kind of a mottled kind of coloration. So this big A allele is causing that little stripe to appear on the fur so that it's not just a solid color. Here, on the other hand, is a solid colored mouse. It's a black mouse. It's small a, right? So in our model system, you have 2 small a's, you're not agouti, you allow the solid coloration. If you have at least 1 copy of big A, then you generate that little stripe. Okay, so these 2 are agouti. This would be solid, non-agouti. And we'll talk about this one later, but this is a yellow mouse, and it's due to a different allele, but we'll get into this probably next lecture. Okay, so just some idea what these things are looking like. This is Mickey. This is a yellow mouse also. Okay, so the book sets up a breeding experiment with mice with different coat colors. And I want to go through this before we end today. Again, it's set up in a particular way to give an illustration of the mechanisms that are occurring to generate the different coat colors. So we have a mouse up here which is agouti, and its genotype is given to us as dominant homozygous big A and dominant homozygous big C. That means Coloration is allowed because you have at least one copy of big C. It means you're agouti. So you can see the mouse looks kind of intermediate, right? So that's agouti. This mouse is albino. It has, it's homozygous recessive for small c, and so no coloration is allowed, so it doesn't really matter what it is at these other two loci. This one happens to be small a, so if coloration was allowed, it would not be agouti. Now what's not shown here is what is this mouse at that B gene. It could be big B, or small b, or something like that. We're not shown the answer to that as the problem is set up here. But once we go through the entire breeding experiment and get to the end, we can evaluate or predict what it must have been at that B gene as well. Okay, so we cross these. What do we get? Heterozygotes at both the A gene and the C gene. One good copy of C allows coloration. One dominant copy of A causes the agouti yellow stripe phenotype. So this F1 looks just like this parent. These traits have been lost, just as we've seen in those early Mendelian experiments. And as always, we take the heterozygote F1s, we cross them together. We say that we can treat A and C separately because they're on different chromosomes. So we do this whole rigmarole again. 3/4 big A something, 3/4 big C something, 3/4 big A, 1/4 small c, and so on and so forth. You get the idea. It's the same thing over and over and over again. And what do you get? 9 parts dominant at both, 3 parts dominant at one and not the other, 3 parts dominant at the other but not the first, and 1 part recessive homozygous at both things. And now we have to kind of collect the output and see what ratio we get. So the rules were, if you're big C, coloration allowed, big A, agouti. So there we get 9 parts this. The rules are, if you're small c, you disallow coloration regardless of what you are at those other loci. So here's 3 parts small c, but this one is also small c. So now we get 3 + 1 gives us 4 parts albino. And this one right here is coloration allowed, but no agouti. Therefore, it must reveal whatever is present at that B gene, which we were not told up here. And you can see that this one is black. So therefore, we could figure out that it must have been big B at that B gene locus, even though we weren't told that here. You could work through this and imagine other possibilities: big B, big B, small b, small b. Other possibilities might occur for these mice at the beginning, and you're going to get different outputs at the end as a consequence of those alleles at the B locus. But in this case, we just get this black, so we know kind of what it is. Okay, so what do we get? We get another ratio. Still, out of 16 boxes, 9-3-4. Something else different entirely compared to the Mendelian things that we looked at before. Okay, so last thing today, 1244. Last thing today is just to say that the genetics of coat coloration in mice seems to transfer to coat coloration in other organisms, and here's some just some front pages of various web pages having to do with dog breeders, particularly Labradors. And Labradors come in like the black lab, chocolate lab, yellow lab, so on and so forth. And some of the genetics for dogs matches the genetics in